Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 4 for June 2023. Let us begin with the first question. Question 1 says, a student investigated the uptake of magnesium ions by some plant cells in the presence and absence of oxygen. The graph shows the results of this investigation. So on the horizontal axis we see time, and on the vertical axis we see the magnesium ions taken up by the plant. So without oxygen there is less magnesium ions taken up, while with oxygen there is more magnesium ions taken up. So maybe that means the process of taking up these magnesium ions requires oxygen, and therefore it's going to be aerobic. So they say, explain the importance of magnesium ions to a plant. Magnesium ions are used in making chlorophyll, and this chlorophyll is going to be used in the absorption of sunlight that is useful for photosynthesis to take place. So I said plants use magnesium ions to make chlorophyll, and chlorophyll is used to absorb sunlight. In part B, the student suggested that magnesium ions are taken up by active transport. Explain why the student came to this conclusion. They want you to use the information in the graph to support your answer. This is true because we saw from the graph, let me take you back, there was more magnesium ions uptake in the presence of oxygen, and that means this process is going to be more aerobic, involving active transport, because respiration occurs, or aerobic respiration occurs, ATP is produced, and that is going to be used in active processes. So here I said more magnesium ions are taken up with oxygen than without oxygen. Also, the magnesium ion concentration levels off without oxygen but doesn't with oxygen. Again, to take you back, you can see this is continuing while this is leveling off without oxygen. So this shows that magnesium ion uptake can occur against concentration gradient. Since oxygen is required for aerobic respiration that leads to ATP synthesis, this shows that the uptake of magnesium ions requires ATP. Uptake with ATP can only occur by active transport. So this brings us to the end of question 1. Let's continue to question 2. In question 2, the photograph shows a black bear. They say most black bears have a diet consisting of ants and berries and hibernate during the winter months. One very large male black bear called Hank was suspected of raiding nearly 40 homes in the Lake Tahoe region of America for food. Some of these raids took place during the winter months. Hank weighed 227 kilograms, which is 1.7 times more than the mean mass of a typical large black bear and 5 times more than the mass of a typical small black bear. They wanted to calculate the difference in mass between a typical large black bear and the typical small black bear. So the mass of a large black bear, because they say it is 1.7 times more, so this is going to be 227, which is the mass of Hank divided by 1.7, giving us that, which is approximately 133.5 kilograms. And the mass of a small black bear is going to be, because they said 5 times, so we go with 227 divided by 5, giving us 45.4 kilograms. So the difference between them is going to be that minus that, giving us 88.1 kilograms if you round off to 3 significant figures. So my answer was 88.1 if you are to 3 significant figures. The next they said, explain why Hank was much larger than typical black bears. Now remember, from the introduction they said Hank raided homes and stole food. So because he stole food from the raided homes, these foods contained a lot of nutrients like proteins. And again, proteins contribute to the mass. So proteins, carbohydrates, and these enable the bear to have more biomass, and therefore it was able to store down some as fat, leading to the increase in size. In part B, samples of DNA were collected from the homes that Hank was suspected of raiding. Analysis of DNA showed that some of the raids were by 2 unrelated female black bears, which 2 procedures could have been used to analyze the DNA. Of course, here they have to do PCR followed by gel electrophoresis. PCR is going to amplify the DNA, and then gel electrophoresis is going to be used to compare the bands. Next, they said, describe how the analysis of the DNA would show that these 2 other bears were female and not related to each other or to Hank. So if their females are not related to each other, it means if we are to use gel electrophoresis in order to compare the bands, of course, they will have bands matching to the DNA of females, but also they will have bands that are not similar to all of them because they are not related to each other as well as to Hank. So I said, using gel electrophoresis, the produced bands could be compared to observe similarities and differences. Observe the band size, the number and position along the gel. Because the females are not related to each other, their band patterns will be different. The band specific to females will be present in the two female bears but missing in Hank's DNA. So this brings us to the end of question 2. Let's continue to question 3. Question 3. Tetracycline is a bacteriostatic antibiotic and vancomycin is a bactericidal antibiotic. They want you to compare and contrast bacteriostatic antibiotics with bactericidal antibiotics. So if a question asks about comparing and contrasting, you have to give the similarities as well as the differences. So I said both antibiotics work against bacterial growth. Of course, they work against bacterial growth, but in different ways. These ones here destroy or kill the bacteria, while these ones here prevent the increase in the number of the bacteria. So my next point was bacteriostatic antibiotics prevent bacterial growth by inhibiting cell division, while bactericidal antibiotics kill the bacteria. Moving on to part B, antibiotics can be identified using RF values obtained from chromatography. The table shows the RF values for tetracycline and vancomycin obtained using two different chromatography methods. Tetracycline with method 1, we have 0.51, and method 2, we have 0.75. And of course, these different RF values are maybe because they use different solvents. So in method 1, if they use a different solvent, we're going to have a different RF value if a different solvent is used in method 2. Then for vancomycin, we have 0.09, which is with method 1, and 0.29 with method 2. So next they say the diagram shows a chromatogram for one of these antibiotics using one of the methods. This is the origin, and this is the point where the sample has traveled. So here they say describe how this chromatogram could be produced. This is easy. You have to get a chromatograph paper, draw the baseline, and then pour a suitable solvent into a beaker, dissolve the antibiotics into a suitable solvent, and then put the antibiotic mixture onto the baseline. The baseline is this line here, which we call the origin. So you mix the antibiotics and then put here so that you allow the solvent to carry the substance upwards as the solvent moves from the origin towards the solvent front. Then you dip the chromatograph paper into the solvent, ensuring that the baseline does not touch the solvent. Then cover the contents to prevent evaporation, because most solvents used are usually volatile. Allow the solvent to move up the chromatograph paper until the solvent front, which is near the end of the paper. In the next part, they say which identifies the antibiotic in the diagram and the method used. Measure the distance from the origin to the sample and then divide that by the distance from the origin to the solvent front. So when you do that, I get approximately, which is 0.71, and this 0.71 is closer to 0.75. If you go back here, 0.75 is going to be method 2, closer to method 2. So my answer was tetracycline using method 2. Next, they say explain why the 4 values in the table are all different from each other. Of course, like I already said, they're different because different solvents were used. So the difference in RF values is because different solvents or different chromatograph papers were used. The antibiotics have different solubilities in different solvents. Tetracycline is more soluble in both solvents than vancomycin. The reason for that is tetracycline had higher RF values in comparison to vancomycin, and that means tetracycline traveled further away from the origin in comparison to vancomycin. So that was my last point here, emphasizing the idea that tetracycline was more soluble in both solvents in comparison to vancomycin. So this brings us to the end of question 3. Let's continue to question 4. Question 4, the photograph shows a young giant panda feeding on bamboo. So this is the panda. They say giant pandas have evolved the ability to eat bamboo. Genes that code for enzymes that digest the cellulose in the bamboo are not present in the genome of the giant panda. The giant panda depend on microorganisms in their intestines to produce enzymes that can digest the cellulose. Which of the glycosidic bonds are broken in the digestion of cellulose? In cellulose, we have only beta-glucose molecules, so it's only the beta 1,4 glycosidic bonds that are going to be broken. In part B, the microorganisms in the intestine of giant pandas include both bacteria and fungi. Fungi are eukaryotic organisms. They say put one cross in the appropriate box in each row to show if genes are found in each of the structures in these microorganisms. Of course, mitochondria, genes are found, but in this case, only in fungi. And then for the nuclei, yes, we have only in fungi. Bacteria do not have a nucleus, so they do not have nuclei, so it's going to be only in fungi. in 70S ribosomes. There are no genes in 70S ribosomes, so here we can conclude neither in bacteria nor fungi. In part C, in one study, the succession of bacteria in the intestine of developing giant panda was determined. 3 stages of the development were studied. They are cubs, young adults, as well as adults. The graph shows the relative abundance of 4 types of bacteria in 3 developmental stages of the giant panda. So here we have Streptococcus, which is one type of bacteria. We have Escherichia and Shigella, which is the second type. And then we have Clostridium, which is the third type. Here the highest abundance is going to be in cubs. Here it's going to be in the young adults. And finally here it's going to be in the adults. Which sequence shows the succession of bacteria during development in a giant panda? So since here we have the highest concentration or highest abundance in the cubs, we can say Streptococcus will come first. This is going to be the second, and that is going to be the third because it's present more in the adults. So D ended up becoming my answer. So here they say describe one method that could be used in this study to determine the relative abundance of Streptococcus in giant panda. So here, whichever method you decide, you have to make sure you use aseptic techniques. Because these bacteria are inside the panda, the easiest extraction method is going to be using their fecal material. And then you could carry out dilution plating using serial dilution. You ensure proper mixing after every dilution to ensure that the concentration is uniform. Then use a suitable selection method on each petri dish, like antibiotics to prevent contamination. This is because you're only interested in specific types of bacteria, not everything, so specific selection methods could be used, like antibiotics. Count the number of colonies per agar plate to determine the total number of bacteria in the sample. Then determine the relative abundance by calculating the percentage of Streptococcus as below. It's going to be number of Streptococcus divided by the total bacteria times 100. This will give you the final result. So this brings us to the end of question 4. Let's continue to question 5. Question 5. In an investigation, scientists attached a molecule of BPF to the cell surface of a species of green algae, which we see here. They say these algae are photosynthetic organisms and BPF absorbs green light. This means it's going to increase the amount of light absorbed. The scientists measured the production of oxygen by C. pyrenoidosar and C. pyrenoidosar with BPF attached and BPF only. The graph shows the results of this investigation. If oxygen is produced, it means photosynthesis is being carried out. So down here we have the graph showing the time in seconds on the horizontal axis and the production of oxygen on the vertical axis. With C. pyrenoidosar with BPF attached, we see there is a higher amount of oxygen produced per time in comparison to just C. pyrenoidosar as well as BPF only. BPF attached to the pyrenoidosar is going to allow for more absorption of sunlight, thereby leading to more photosynthesis, and therefore oxygen production is going to increase. Down here they say calculate the rate of production of oxygen at 1,200 seconds for C. pyrenoidosar with BPF attached. So I came here, this is 1,200 seconds, and I drew a tangent. So I used the tangent to find the gradient, which is going to be the rate. I chose to point that point there as well as that point there. You can see the coordinates I used. Let me make it bigger. We have horizontally 1,440, vertically 165, and 880, vertically 125 for that specific point, which is that. So my rate is going to be the change in y divided by change in x. So it's going to be 165, which is that, minus 125 divided by 1440 minus 880. And my answer was 0.0714, which is to 3 significant figures. Moving on. They say explain the effect BPF has on the production of oxygen. So I said presence of BPF on C. pyrenoidosar leads to production of more oxygen. And again, this is because this allows for more absorption of sunlight, more photosynthesis being carried out, more oxygen being produced. So this is because more light is absorbed due to absorption of more green light. This leads to increased photolysis of water, resulting into the production of oxygen by C. pyrenoidosar. However, with only BPF, very low oxygen is produced because the molecule can't carry out photolysis of water. And again, remember, during the process of photosynthesis, oxygen is produced from the photolysis of water. So this whole thing is about absorption of sunlight, more photolysis of water, more oxygen production if we have BPF attached onto the C. pyrenoidosar. In part B, The scientists also measured the mass of C. pyrenoidosa without BPF attached after a 20-day period. They found that the increase in mass of C. pyrenoidosa with BPF attached was greater than the increase in mass of C. pyrenoidosa without BPF attached. Explain these findings. This is easy. Increase in mass is due to more biomass being made within the species. So that means if there is BPF attached, there is going to be more photosynthesis, there is going to be more glucose produced, there is going to be more converted into stored biomass. So I said presence of BPF leads to a faster rate of ATP and reduced NADP formation in the light-dependent reactions. This is again formation of the products that are going to help us in the light-independent reactions. So this leads to a faster rate of GALP and glucose formation. Then more simple molecules are converted into stored biomass in a short period of time, hence the increase in mass. This should be of the microorganism in the presence of BPF. Let me just erase that, hence the increase in mass with BPF. Okay, so this brings us to the end of question 5. Let's continue to question 6. Question 6. Malaria is a life-threatening disease caused by pathogen Plasmodium. Plasmodium is transmitted into the blood of a person through the bite of an infected mosquito. The Plasmodium is a sporozoite that travels to the liver cells. The sporozoite produces merozoites that infect red blood cells. This has stated the meaning of the term infection. Infection is entry of a pathogen into a cell. Next, they say in 2019, there was an estimated 229 million cases of malaria worldwide. The number of deaths was estimated at 4.09 times 10 power 5 per year, which is the approximate death rate from malaria in 2019. So from the information given, we know 4.09 times 10 power 5 deaths occurred out of 229 times 10 power 6. And again, this is millions, so 229 million is equivalent to these patients. The death rate is going to be 229 times 10 power 6 divided by 4.09 times 10 power 5, which is about 600. So 1 in 600 patients died. In 2019, 274,000 young children died from malaria, which is the ratio of the deaths in young children to the deaths in other age groups. From the information given, these are the children who died. Well, this is the total number of people who died per year. So to get the number of other age groups that died, it's going to be the total deaths minus the deaths from children, which gives us 135,000 patients, or I would say deaths. So the death ratio is going to be young to other groups. It's going to be, and actually to clarify here, let me write deaths. That makes it clearer. So the ratio is going to be 2:1. Moving on. In part C, the scientists are trying to develop vaccines to protect against malaria. One vaccine uses living sporozoites, suggests why sporozoites are used in a vaccine against malaria. For any vaccine, the key thing is to trigger the immune system so that memory cells can be produced. If secondary infection occurs, then the immune system is ready to produce antibodies that can be able to carry out the destruction of the microorganisms quite efficiently. So I say it because sporozoites trigger the immune system to effectively produce memory cells against sporozoites. On actual infection, the memory cells rapidly produce antibodies to destroy the sporozoites before they become merozoites. This prevents infection of liver cells and red blood cells. In clinical trials of this vaccine, people were injected with the vaccine and two chemicals. One chemical kills sporozoites and the other chemical kills merozoites. Deduce why these two chemicals are injected in the vaccine in these clinical trials. One chemical is going to be killing sporozoites, but if some survive, then it means they're going to be converted into merozoites, and these merozoites are going to be killed by the second chemical in order to prevent the infection of the cells. Since the vaccine uses living sporozoites, they have the same antigen. However, the immune system takes time to be activated. The sporozoites may change into merozoites that can enter the liver cells. So one chemical is used to kill the sporozoites in the vaccine to prevent formation of merozoites. The other chemical kills the merozoites in case sporozoite killing chemical was not effective. Next, they say, later in the clinical trials, people were infected with one of two strains of sporozoites 3 months after the vaccine. One strain was identical to the one used in the original vaccine, and the other strain was a different one. The trial found that the vaccine was effective against both strains. Explain the findings. If a vaccine is effective against all strains, it means they have the same antigen that is going to be detected by the immune system. So I said both strains have antigens similar to those used in the vaccine. Since memory cells are already present, antibodies are rapidly produced for secondary immune response to occur. So agglutination, opsonization is going to occur quite efficiently. I'm talking about for the sporozoites, they're going to be engulfed by the macrophages in order to be destroyed. So this brings us to the end of question 6. Let's continue to question 7. In question 7, commercial whaling has resulted in a large decrease in the number of whales. The diagram shows the relationship between whales and some other organisms in the ocean. So they say when whales come to the surface, they release nutrients into the water in their feces, and then the phytoplanktons are photosynthetic microorganisms that grow at the surface of the water. The zooplanktons are animals that feed on phytoplanktons, so these act as food for these, and then the fish and other animals feed on the zooplanktons. So we have a food chain here. It means if these phytoplanktons are photosynthetic and the whale releases nutrients into the water, it means the feces of the whale are going to provide nutrients for the phytoplanktons, which are photosynthetic, to grow so that more zooplanktons have food. And once these are increasing in size, there is more food for the fish as well as other animals. And they say when whales die, their bodies sink to the bottom of the ocean where there is less oxygen. If there is less oxygen, there is going to be less aerobic respiration, so less decomposition. And another thing, the nutrients from their decomposition will not come to the surface. When whales dive, they bring nutrients to the surface. So they dive and bring nutrients. The nutrients on their fecal material are going to aid the phytoplanktons in order to grow. Here they say in part A, whale feces add nutrients containing nitrogen to the water. There are 345 right whales in the Gulf of Maine, and together they release 15.9 kilograms of nitrogen per day. Calculate the mass of nitrogen released by one right whale in a year. If together they release 15.9 kilograms of nitrogen per day, it means a single whale is going to release 15.9 divided by 345, because 345 is the total number of whales, so this is going to give us the mass per whale per day. And then since a year has 365 days, so if this is the mass per whale per day, times 365 gives us the mass per whale per year, which is 16.8 kilograms per year if you round off to 3 significant figures. Next, they say nitrates contain nitrogen. Describe the importance of nitrates to plants. Of course, this is easy. Nitrates are going to be used to make amino acids, which in turn are used to make proteins. These proteins can act as enzymes, hormones, or structural proteins. Also, nitrates are used to make nucleotides for DNA as well as RNA. Nitrates are used to make ATP for cellular activity, like in photosynthesis. In part B, there are 1.3 million whales in the ocean. Scientists believe that if the number of whales could be restored to their original numbers, this would have a significant effect on climate change. There were approximately 5 million whales before modern commercial whaling. Calculate the percentage decrease in the number of whales due to commercial whaling. So the decrease is going to be 5 times 10 power 6, which is 5 million, minus what we have, 1.3, which is 1.3 times 10 power 6, that's 1.3 million. This gives us 3.7 million. So the percentage decrease is going to be the decrease divided by the original times 100, which gives us 74%. Next, they say calculations have shown that if numbers could be restored, 1.7 times 10 power 9 US tons of carbon dioxide could be removed from the atmosphere per year. A US ton is equivalent to 907 kilograms. Calculate the mass of carbon dioxide in kilograms that could be removed from the atmosphere in 1 year. They want you to give your answer in standard form. This is the amount of carbon dioxide that is going to be removed per year, and this is equivalent to 907 kilograms. So in total, we have 1.7 times 10 power 9 times 907, giving us 1.54 times 10 power 12 kilograms per year, and this is my answer. Next, they say discuss the possible effects of increasing the number of whales on climate change. Now, remember they've told us that the whales remove CO2, so there is going to be a decrease in CO2. There is going to be more whale feces and therefore more nitrates, meaning the phytoplanktons are going to have more biomass, and that biomass is going to feed more zooplanktons, and then the fish and other organisms in the water are going to have more food as well, so there is going to be more growth. But with more growth, there is more death and more decomposition, So they say use the information in the diagram and the data given in this question to support your answer. I said whales dive up the surface and they introduce more nutrients from their feces. These are a source of nitrogen to the phytoplanktons. The phytoplanktons increase in number and carry out more photosynthesis, leading to the decrease in CO2 levels. Excessive growth of phytoplanktons leads to abundance of food for other animals like zooplanktons. When they die, their decomposition leads to more CO2 release back in the atmosphere. This contributes to global warming. Whales consume large amounts of food. They act as carbon sinks, meaning they take up a lot of this carbon, and remember, when they die, they sink to the ocean. The rate of decomposition is slower due to the conditions, so the carbon dioxide produced will remain in the ocean, and this reduces on global warming. So this brings us to the end of question 7. Let's continue to question 8. Question 8. Reforestation is an example of effective management of conflict between human needs and conservation. Organizations have put together a list of rules for responsible reforestation. These rules aim to improve absorbing and storing atmospheric carbon dioxide, biodiversity, and human livelihood. 4 of the rules are: number 1, protecting existing forests first, work together with local people, select appropriate areas for reforestation, and select species to maximize biodiversity. Explain the importance of protecting existing forests, which is the first rule. Protecting the existing forest helps us to protect the tree species that are already existing. So to protect the existing tree species, maintaining their biodiversity and continued carbon dioxide fixation. To protect other species that rely on available tree species for food and shelter, of course, these are animals as well as birds. Also to protect the land from soil erosion, because these roots, or the tree roots, hold the soil together. And again, this is all for the already existing forest. You have tree species that are already there. These need to be protected. You have the animals and organisms that depend on those specific tree species. Their food as well as shelter has to be protected. And again, the soil has to be protected from erosion. In part B, suggests why it is important that organizations involved in reforestation work with the local people. The local people depend on the forest or on the land for survival, and if they're not educated, there is a chance that the reforestation effort may not be successful. Because the local people depend on the land for food and a source of income, however, the local people need to be educated about the side effects of cutting down trees. Working with the local people enables them to see the benefits of reforestation to the climate. In part C, explain 2 reasons why previously forested areas should be selected for reforestation instead of other wild areas. We do not have to destroy land that is already occupied or land that is used for farming in order to plant these trees. If wild areas are used, other ecosystems will have to be destroyed as land is created for reforestation. This could lead to species decrease or extinction, decreasing biodiversity. If farmland is used, then people will have less land for farming, less food supply, and farmers who depend on the land to earn a living will be out of work. Also, previously forested areas are already capable of maintaining certain tree species. They could still have some seeds, and this leads to faster reforestation. In part D, explain why a mixture of native species, including endangered and genetically diverse species, should be introduced. This is the 4th rule. Now, native species have previously thrived in these areas. Those that are endangered need to be protected because they could become extinct. And genetically diverse species, these can withstand extremities of conditions, like if there is selection pressure, they can be able to withstand the harsh conditions. So I said native species have previously thrived in the area, so they are capable of withstanding the conditions and acting as a source of food and shelter for local animals as well as birds. Including endangered species decreases the chances of those species from becoming extinct. Including genetically diverse species increases biodiversity and the gene pool, and this ensures survival if the environmental conditions change. So this brings us to the end of question 8. Let's continue to question 9. In question 9, the time of death of a mammal can be estimated in a number of ways. Part A says, measuring the body temperature of a corpse is one method used to estimate the time of death. The graph shows a calibration curve that can be used in this method. On the horizontal axis, we have time after death, and on the vertical axis, we have the temperature of the corpse. We see the temperature of the corpse is decreasing as time after death increases. The first part says describe how the time of death of a person can be estimated using this method. We're going to obtain the core body temperature, and then we will come and match the temperature to the graph that has been drawn to us so that we can estimate how many hours after death. So I said by inserting the thermometer in the rectum or liver of the corpse to determine the core body temperature, then you refer to the graph at the core temperature to estimate the number of hours after death. You then subtract the hours after death from the actual time the corpse's core body temperature was measured. Next, they say explain the limitations of this method. Of course, we know the temperature of the corpse is affected by many different conditions. If we are using core body temperature, the ambient temperature as well as the way the body is formed is going to affect the core body temperature. For example, if the body is clothed, then temperature is going to be lost slowly. If the person is fat, temperature is going to be lost slowly. But if the body is in a colder place, then the temperature is going to decrease rapidly. So here it said after death, metabolism stops, so heat loss occurs. Heat loss depends on factors like ambient temperature, body fat content, mode of death, and more. Corpses with more fat lose heat at a slower rate. If the ambient temperature is low, faster heat loss occurs, and then a corpse with open wounds loses heat faster. In Part B, scientists are looking for other methods to estimate the time of death using presence of chemicals in the blood. One study used blood taken from 5 rat corpses and 2 pig corpses at intervals after death. So just why this study used rat and pig corpses instead of human corpses. Of course, it's unethical to use human corpses in this kind of studies. Next, they say the graph shows some of the results of this study. So we can see on the horizontal axis, we have time of death in hours, and on the vertical axis, we have the pH. Here we see the pH decreases as time of death increases. And this is because, of course, there is anaerobic respiration stalls, more lactic acid, the pH is going to decrease. And then the next graph, time after death, as time after death increases, the levels of ammonia in blood increase. Next, we see time after death in hours as it increases. The levels of hypoxanthine in blood, we see it increases over and then decreases, but of course there is overlap. And then lastly, the levels of lactate in blood, as time after death increases. Overall, there is an increase. Error bars were placed, which means the experiments were repeated, but there is some overlap. Even at different times of death, like 9 hours after death and 24 hours after death, we see there is overlap between these. So here they say comment on the usefulness of using these chemicals for determining the time after death of rats and pigs. Use the information in the graph to support your answer. So I answered this one graph at a time. So I began with a pH graph. Overall, the pH of blood of both rats and pigs decreases with increase in time after death. Error bars show overlap, so the results are not valid. There is a shorter error bar for pigs than for rats, so the results for pigs are more valid in comparison to those for rats. When we go to the levels of ammonia, initially there is more ammonia in rats than pigs, but after 24 hours there is more. This should be more. There is more ammonia in pigs. No overlap for pigs, so the results are valid. With hypoxanthine, overall there is an increase in the chemical in both rats and pigs until 32 hours. Let's see. Yep, until 32 hours we can see it's increasing until 32 hours. There is more chemical in the rats than pigs until 48 hours. However, no visible trends in data because it goes up and down. Looking at lactate, up to 6 hours, there is more lactate in the rats than pigs. The error bars for pigs and rats overlap. The data for pigs has shorter bars than rats, so it is more accurate, but there are no clear patterns from the data, so the data is not useful. In my conclusion, overall, the sample size was small. They used just 5 rats and 2 pigs. The corpses are not representative enough, and the results are not that significant. Lastly, they say discuss whether these chemical tests are likely to be useful in determining the time of death of the human. So pH after death, I said pH after death decreases. This is true even for humans, it's going to happen. So pH data is similar to humans, and that can be useful. Humans and these species, which are the rats as well as pigs, could produce different chemicals after this. So this could be misleading. If the rats and pigs produce some chemicals, the humans could produce different one. So testing on human corpses is better to conclude on the suitability of these chemicals. Like I said already, the sample size is just 5 rats and 2 pigs. This is not representative enough. they need to use more animals, and there is a lot of overlap, so the results are not valid, and therefore this may not be useful in humans. So this brings us to the end of question 9, as well as to the end of this whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.